Good morning, good afternoon, good evening to all of you. Join us today for our Going Organic Seminar Series for the spring 2022. My name is Dil Tavaraja. I'm an Associate Professor leading the Pulse Quality and Organic Nutritional Breeding Program. Today we have very interesting talk from Dr. Anne-Marie Fortuna. Before we go there, I first like to thank our sponsors. The United States Department of Agriculture National Institute of Food and Agriculture Award. 2 awards we have from NIFA OREI, both for pea and lentil organic breeding. And USAID Feed the Future Innovation Lab, Cornell sub-awardee. And South Carolina Department of Agriculture, the Good Food Institute, and the Food Shot Global. I would like to thank all our sponsors for helping us to get this program running and having these wonderful speakers every month on the organic breeding and the management. Now I'd like to introduce Mark Dempsey, one of my PhD students, also one of the managers at the Carolina Farm Steward Association. He holds 2 hats, and he is our moderator today to work with Dr. Anne-Marie Fortuna. And Mark, this is your floor. Thank you.
Bill, it's my pleasure to introduce Dr. Fortuna. Dr. Fortuna's research focuses on microbial and soil processes, regulating nutrient cycling, soil health, and global climate change in agricultural and grassland systems, as well as the use of soil health indicators to measure the effectiveness of remediation and land management strategies. Her nationally recognized program addresses environmental quality and its impacts on land, air, and water resources. And we're really excited to have her here today. So with that, I'll hand it over. Thanks a lot.
Thank you. Lovely introduction. So I'm going to present 3 different projects today, and they all use the same techniques that are biological indicators of soil health. And the interesting thing I think about organic systems is that many times things that are tried on these small areas, relatively speaking, to the tenths to hundreds of hectares that some production agriculture is in, make it easy to change and be dynamic. And so we've seen a lot of technologies go from smaller organic farms to larger operations. And so the first 2 projects are sustainable, but not organic. And then I'm going to end with an organic project. So with that, I will begin. And so the rationale and objectives I just covered. So we're looking at a set of biological indicators, specifically the nitrification pathway, and in particular, using gene copies of nitrifiers to assess.
Dr. Fortuna, you need to share your screen.
Oh, sorry, I had done that previous. Okay. Oh, I see. For the introduction, we must have not done that. Thank you.
If you can go to your first slide or, yeah.
Yeah, I'm on the second slide.
All right.
Can you see that, Del? Yeah, okay.
Yes, we can.
Using the nitrification pathway, and in particular, the nitrifier gene copies, which is a molecular technique, which is fairly simple to assess nutrient cycling remediation strategies. In this instance, it's going to be related to manure and manure byproducts, and also the nitrification pathway's influence on greenhouse gas emissions. So I just want to briefly touch base on what nitrification is. I know many of you already are familiar with it. Essentially, it's the conversion of ammonium to nitrate. And organic systems are unique in that most of the nitrogen is bound to carbon. And so it goes through a process of what we call ammonification, release of nitrogen from that organic carbon, and then gets converted fairly rapidly from ammonium to nitrate. And there's only 2 organisms that we know of in agricultural systems that can nitrify: ammonia oxidizing bacteria, which we've known about for a very long time, and ammonia oxidizing archaea. Both are single-celled organisms, and they are they occupy different niches or ecosystems. And so we'll talk about how, because they are in different niches, they can give us some information about our inherent properties, which is related to soil health. That's things like our soil texture and mineralogy, and also gives us an idea of how our land use and best management practices influence nutrient cycling and their potential to nitrify. And so because there are a number of intermediate pathways that are less clear as to what transformations are going on, this pathway is, the conversion of the nitrite to the nitrate is a part of the pathway that only goes in one direction. It's limited by the energy required to transform nitrite to nitrate, and that's controlled by a set of enzymes that these ammonia oxidizing organisms have. And so that's what makes them a really good indicator. And you can see that one of the byproducts of that conversion process can be nitrous oxide. So there's a link to greenhouse gas emissions as nitrous oxide through this nitrification pathway. And then denitrifiers use the end product, the nitrate, in the process of denitrification. So there's an additional linkage there. So now that you've got a little bit of biochemistry, let's move forward. All three of these projects use similar techniques. So we looked at plant available nitrogen, ammonium nitrate. This is a KCl extract any soil testing lab can perform. The nitrification potentials are in the laboratory, and essentially what they represent is the maximum potential of these ammonia and archaeal oxidizers on a given day. So when I take my soil sample, I incubate them, and at that point in time, that's the most nitrification they're more than likely to produce. And so that keeps things under a standard set of conditions, under ideal conditions specifically. And then using the gene copies, both the DNA and the RNA are helpful because these organisms are a little slower growing. They generate in about a week under ideal conditions, and they have about 1 to 3 copy numbers of this gene in their genome. And so you're measuring almost one cell per reaction. So it's not a true biomass measurement, but we can equate it somewhat to the biomass increase or decreases of both ammonia oxidizing bacteria and archaea. And then I use some just very basic statistics to analyze the data, Pearson correlations, and then this PROC mixed routine to do standard deviations and means. So the first project is interesting. I mean, it was a national project that I conducted when I worked for the USDA the first time. We took benchmark soils. So benchmark soils are defined by Natural Resource Conservation Service, and they represent a large acreage of a given region. And so they have all the background information about these types of soils. And we thought using them would be useful to growers and to the public because they would represent a larger amount of the surface area. So we looked at nitrogen cycling and specifically the nitrifier communities and the nitrification potential. And we looked at how different mineralogy interacted with the microbial communities and how ammonium from dairy slurry was integrated into the mineralogical and biological components. So we had 9 soils. I'm just going to pare it down to 3. The dairy slurry manure contained 170 kilograms of ammonium and then another 130 kilograms of organic nitrogen. So over a 30-day incubation, we had the 170 kilograms immediately available and a portion of the organic nitrogen ammonified and converted to ammonium. So this table contains the 3 soils. So the take home is that all clay is not equal. So for a given amount of clay, say 400 grams per kilogram, if we have a high shrink swell clay with high cation exchange capacity, we'll get a different amount of loss or not loss of ammonium due to volatilization. and we'll also see a different type of interaction of the ammonium with the surfaces and the inner layers of these clays. So the first two soils have the same amount of clay. They're the same texture, silty clay loams, but their mineralogy varies. So the Catlin soil from Illinois is dominated by smectite. Smectitic clay allows for absorption of ammonium in inner layers, and it can be exchanged back into the soil solution because the layers are popped open. So in the Sharpsburg soil, where illite is the dominant soil, we have inner layers that are tighter, so you can get immediate fixation and sorption into the inner layers, but you don't get it coming back out. It's fixed. And potassium is another iron that often gets fixed in illite and is on the edges. So these two soils also have some variation cation exchange capacity as well. And the cation exchange capacity on the outer layers, the most readily available ammonium, it exchanges immediately if you have a decrease in ammonium in the solution adjacent to the mineral. So this is how plants and microorganisms mine ammonium from the soil. And then finally, the third soil has only 60 grams of clay per kilogram. The texture is a sandy loam, but it has an equal amount of illite and smectite. So I wanted to see how the combination of the two minerals together influenced the availability of the ammonium and how that related to nitrification potentials and possibly copy numbers of ammonia oxidizing bacteria and archaea. So again, ammonia loss is a really big issue, especially when we're applying animal waste or organic materials, because oftentimes we get a modification or there's ammonium there and it can immediately volatilize. And under the ideal pH conditions of about 7 to 8, a pH of 7 to 8 would be ideal for loss, ammonia volatilization loss. And so I think what's going on in these minerals is in the smectitic dominated soil, which is that blue bar, we see time 0, 7, 14, 21, and 28 days of the incubation. We plotted that against the potential for nitrification. So the smectitic clay more than likely is immediately sorbing the ammonium, and then it's slowly releasing over the entire incubation. You see that from day 7 to 28, you just have this steady supply probably of ammonium, which makes the nitrification potential constant. If you look at the second bar, the yellow bar, the illite's not easy to read, but that's the soil that had the same amount of clay, but it was dominated by the illite. So you see probably volatilization, a combination of volatilization and fixation in the inner layers on the illite. And you see an increase between day 0 and day 7, but it's about a third to a half that of the smectitic dominated soil. And so you also get a constant amount of ammonium that's available for nitrification, but much less. If you look then at the last bar in green, that's the soil that had only 60 grams clay, but it had the mixture of illite and smectite. You see an interesting trend. There's this slow, gradual increase from day 0 to 21 in nitrification potential, and then a drop on day 28. So you're definitely getting a mix of the two, and they're reacting similarly to the soils where they're separate. And actually, you're getting more nitrification potential in the mixture that has less clay. So you can see that the mineralogy really has a big impact on nitrogen use efficiency. And in the organic systems, that's really the name of the game, is to try to figure out how things are mineralizing and releasing for plant uptake, ideally in the window of maximum uptake for whatever crop you're growing. So now this is a shift into the communities. It's the bacterial ammonia oxidizing bacteria. And there's 3 groupings. The first is that soil dominated by the smectite. And the second is got the soil with the dominant illite mineral, same amount of clay, but you can see that the potential for the soil that has the smectite to essentially provide nutrients longer term to the ammonia oxidizing bacteria is greater for the same amount of clay than in the soil with the illite that fixes the nitrogen. So it's like it's like if you're putting a feeder out for an animal, and it takes some food out and then more drops down. And then with the smectitic illite combination, we get a more similar trend to the soil that was dominated by the illite. So you can only transform, so much material at a given time. But bacteria are interesting in that they can be static. And then when they see a substrate, they can just ramp up their metabolism. And so that's likely what's going on here, is that we're getting organisms that were not active suddenly, or at, displaying a lower metabolism increasing. Now we're moving to the archaeon. Again, we see a similar trend where presence of smectite seems to make the potential for nitrification increase, and also the populations increase. And so here there's a statistically significant difference between the second soil dominated by illite and the mixture. But I would say, that it obviously hasn't had a positive or negative impact on nitrification. Okay. So basically in the first project, the inherent soil properties are what drove the nitrification rates and the potentials. We're moving to a second experiment that was conducted by one of my students. It was a very interesting experiment. We had an in-field and laboratory component. We had a relationship with Dairy Gold, which is a very large cooperative that sends dairy solids and milk all around the world. They decided that they wanted to treat their waste environmentally, and they wanted to build a bioreactor. And there was a plant that had food waste that also needed to be put into some kind of environmentally friendly stream. And they combined these two waste in an anaerobic digester that produced methane for the local town. And so we took the raw material from the dairy, they call it toilet water, and then we took this waste that was generated from the anaerobic digester and compared that to urea fertilizer. So the idea is that we wanted to have this environmentally friendly farming ecosystem where we could take the byproducts of this very large dairy and make it sustainable. And so again, we're using the bioindicators, the ammonia oxidizing bacteria, the nitrification potential. And in this instance, we're also comparing the sources of nitrous oxide. So we're looking at which communities are active, the ammonia oxidizing bacteria, denitrifiers, or both. So we want to know source, and we want to create some management practices that reduce losses of reactive nitrogen. So ammonium, nitrate, and nitrous oxide. So I just want to remind you that the pathway of nitrification, one of the byproducts is nitrous oxide. That's why I'm showing you the slide again. And that ammonia goes into the nitrification pathway, and then we get nitrate produced, and nitrate is the beginning of denitrification. And so we see the pathway for denitrification in this slide. There's the large circle, and the first component is nitrate, and then it's getting converted all the way down to nitrous oxide, and then finally elemental nitrogen. So if denitrification is complete, you get an inert gas that makes up a majority of our atmosphere, elemental N. And so you can control, using your best management practices, up to a certain point, the amount of nitrous oxide generated by keeping the conditions for the denitrifiers in this instance need organic carbon, the nitrate from the nitrification pathway, or if there's nitrate in fertilizer, and then they need a certain redox potential. And if you keep those things optimal, you'll get a minimum amount of nitrous oxide produced. And in this system, the conversion of the nitrous oxide to elemental nitrogen is controlled by the NAZ gene. And that's produces an enzyme the denitrifiers do that allows for that transformation to be energy efficient. So you can see that if you're applying a slurry that has a lot of water, that you could have anaerobic and aerobic conditions, and that could lead to both things happening at the same time. So this is the content of nitrogen of everything that we applied. So the digested manure contained 134 kilograms of nitrogen per hectare in ammonium, and then another 70 kilograms in organic nitrogen that can be ammonified and converted. The dairy slurry contained about half, not quite half, the ammonium, and the same amount approximately of organic nitrogen. And then we applied urea as a control, 140 kilograms of nitrogen per hectare, all in ammonium. So you can see there's different potentials already for ammonia volatilization because there's differences between the two applications. Just when you're applying it, there's almost double ammonium in one. But also we did subsurface and broadcast applications, thinking that if we trenched, that we would get less ammonia volatilization because we would have more contact with the soil surface. So we looked at ammonia volatilization using the same technique in the field and in the laboratory. We created these oxalic acid traps, and it trapped the ammonium, and then we extracted that with KCl and measured the amount of ammonium. And then in the laboratory and field, we also took nitrous oxide samples. So the absolute concentrations were different in the field and lab, but the trends for what was significantly less or greater were the same. So the laboratory incubation does relate to the field. And I'm presenting the laboratory incubation because that's where we did all of our molecular work. I'm not going to talk about the controls that we used. Those are in the paper, and we'll provide you the references. And again, I'm using the same techniques, extracting inorganic nitrogen, looking at the same genes in the AOB, except for I'm using RT-PCR. So I'm looking at the active component. Messenger RNA gets converted to the cDNA, and we can tell how many copies of the organism are present. So it's essentially like a biomass measurement. And then we did the same for the NAZ gene. The NAZ gene is a little more complicated because denitrifiers vary more. They're not as specific a population. So now I'm presenting the actual data. This is the ammonia volatilized from the different applications. We did have a no nitrogen control. That's on the very left. So urea lost the most ammonia. That makes sense because we applied all of the nitrogen. It was essentially converted to ammonia immediately. And there's no way to trench or not trench. If we look at the different slurry materials, the digester is the orange and the red bar, and then the toilet water, the slurry from the dairy that's raw, are the two purples. So we see that we lost about the same amount of ammonia, except for where we subsurface applied the raw from all of the materials. And if you remember, there was more ammonia in the anaerobically digested material. So even though we lost the same amount, digesting the material reduced the amount volatilized per unit of ammonium. So that's one best management practice. If we look at how we applied it, we didn't do so well. And I think our subsurface application still left enough surface area. The soil didn't cover the top of the trench. And so we still had the potential for volatilization. And that was something we could improve upon. Now we're shifting to the nitrous oxide produced. And here we see some radical differences in the digested and raw slurry. So again, the way that we applied it didn't have much impact. Again, here, I think there was enough ponding on the surface that we got anaerobic sites. So the digested material provided or resulted in the most nitrous oxide loss. There were carbon components maybe in that material from the food waste that could have aided the denitrification process, because we need our organic carbon for that. So that was one of the theories we had for the differences. And again, we see that the toilet water, the dairy slurry that's raw, produced about the same amount of nitrous oxide as the urea application. So a lot of people are concerned that when you apply animal materials that you increase your potential for nitrous oxide loss. But in this instance, at least with the raw material, that wasn't the case. So now I'm shifting to the community structure. So the top line is our ammonia oxidizing bacteria. And then the third line is the NOZ. That's that enzyme that converts the nitrous oxide to elemental nitrogen. We're not gonna talk about the middle row, that's the NRG. So the take home for this slide is that we saw no increases in the ammonia oxidizer copy numbers or the denitrifiers where we applied the urea, but we did see increases in both communities where we applied the animal-based products, and how we applied it or whether we digested it or not did not make a difference. So there was an increase where we applied that manure material. And even though there were some differences in the amount of nitrous oxide we lost, there wasn't differences in copy numbers between these communities. So again, we're seeing that the bacteria can either ramp up or down, and that a static population can handle a large amount of input of ammonium and nitrate. Okay, this table correlates the nitrous oxide produced with the copy numbers of both the ammonia oxidizing bacteria and the denitrifiers. They're circled, and there's a strongly significant correlation between the production of the nitrous oxide in both communities. So how I would interpret this is that, yes, we are getting probably nitrous oxide from the nitrification pathway, but we're also seeing that nitrification leads to the production of nitrate, which is the substrate for the denitrifiers. So there's a strong linkage in these systems between the two communities. So this shows that bioindicators do predict, are good predictors. The functional gene copies tell us something about management, but they're not increasing or decreasing directly with increases in production, but they can still be used as bioindicators of inherent and variant management. And so this shows that, that these soil health indicators can be used quickly to assess differences in land management and production. And so, this is something that we could potentially deploy across a large region to answer some questions on a regional or national scale. And I think that's really critical. So now I'm getting into the organic management. So the field plots in this part of the talk, have been in place for over a decade when we did this experiment. And again, we're using these biological indicators of soil health to estimate the fate of reactive nitrogen in long-term organic vegetable and pasture systems. So we had some annual and perennial systems that we were comparing, and we integrated cover crops with animal waste. So we'll look at the correlation between denitrifier and nitrifier. In this instance, I'm only presenting the nitrifier data with the reactive nitrogen species, the ammonium nitrate, and I'm not presenting the nitrous oxide on this one. So the organic systems tend to be very complicated, and I think the easiest way to explain the design of the experiment is that we wanted to integrate green manures, but green manures alone don't give us enough nitrogen. So we needed to integrate animal waste, and we did that. We had broiler hens, and we also had compost that we were making on site. And so what we wanted to do is always deliver approximately the same amount of organic or inorganic N or plant available nitrogen to the vegetable crops each year that we were growing. And we had vegetable crops that had about the same requirement for nitrogen, even though we were rotating them around. So it wouldn't make much of a difference what we were growing. Ideally, we would still have the same management practices and we'd still get the same amount of nitrogen out. So we had the green manures plus a low carbon treatment, which was the broiler litter. C to N ratio is 11:1. We applied 6 megagrams per hectare of material. And then we had a high carbon treatment, which is mixed on-farm compost that still has a low enough CN ratio, 15:1, to release nitrogen. But there was an additional humic fraction that also contained nitrogen that wasn't readily available. We were adding a lot more material in that system, 33 megagrams per hectare. So that had implications for carbon, but that's a different story for a different day. This slide looks at the inorganic nitrogen that was provided. And so the bars, at least the 5 from the last 5 from the left, all provide the same amount of inorganic N. So no matter what treatment we're in, whether we're tilling, not tilling, whether we're adding compost or the broiler litter, we're still getting the same amount of inorganic nitrogen at the beginning of the field season when we're turning things under. The alley was just a check. It was just showed the buildup of the organic material prior to our management. This slide represents the same treatments, but it's the nitrification potential. And here you can see that most of the bars are not significantly different. So the only treatment that shows a little bit more increase in nitrification potential is where we applied the compost. So that shows you that even if your inorganic N, your plant available N is higher, you're still building up nitrogen in your system that's influencing your potential to nitrify, even at times of the year where you don't necessarily need the nitrogen. So we're doing a good job of pulsing the nitrogen out when it's needed and retaining it when we don't have active plant uptake. This represents the June and September field dates. And again, here we're looking at the nitrification potential. So the lay pasture is the perennial, and then the 2 other systems are the annual systems. And we're getting, again, similar nitrification potential on our vegetable cropping systems. And then the lay is going to vary because it's an annual system. September is so high in that system because grasses tend to turn over in fall. And so that represents the pulse that comes out from the turnover of that grass lay. And then we see differences, not much, but to a small degree in nitrification potential between, again, the low and the high carbon management, chicken manure being the low and the compost being the high. But overall, we're matching our nitrogen nicely between these two, these sets of inputs. And so I think that's really where the beauty of organic management comes into play, is trying to integrate all of these different C and P cycles in a meaningful way, and then doing that across multiple rotations, finding the commonality, understanding how we can have consistent best management practices and vary our vegetable crops and other things that we want to diversify with. I don't know if you can see the top of this slide or not. It's ammonia oxidizing bacteria is got the red bar across the top, and the gray is the ammonia oxidizing archaea. So we're looking at the June and September dates. In March, the numbers and the rates were pretty, the numbers of the bacteria and archaea didn't really, were lower, and they didn't give us much information. So we focused on June and September, June being the time where we need the most inorganic N, and September where we're ramping down. So with the ammonia oxidizing bacteria, We don't see differences in the copy numbers given the date, but we do see differences between the low and the high carbon inputs. So where we apply chicken manure, we have less carbon to tie up that inorganic nitrogen. And so we're getting higher copy numbers in where we're applying the chicken manure relative to the compost. And in the compost systems, we have lower AOB. If we shift now over to the archaea, we see that there's no difference in copy numbers depending on the time of year or the two sources of nitrogen. So again, I think that archaea they need very little nitrogen. They're more oligotrophic and they're mixotrophs, so they can use the carbon in the compost as well. So you're not really seeing, they're insensitive to the applications. And if you look down at the Pearson correlation for March, you can see that there's actually a negative interaction between nitrification and the ammonia oxidizing archaea. So this shows that the niche of this community is probably not in a high nitrogen environment. June has the most interesting interactions. There's interaction between total carbon and nitrogen. That's the purple circle at the bottom. That makes sense because in organic systems, all your nitrogen is bound to the carbon and you go through this process of ammonification. So they should be connected. You also see an interaction, a positive interaction, although it's not, it is significant between inorganic nitrogen and total nitrogen in the soil and inorganic nitrogen and nitrification. And then there's a positive interaction between the two groups of ammonia oxidizers as well. So this is the time point where you've got the most inorganic plant available material in your system. And then as you go back to September again, you just get this one interaction, a negative interaction between nitrification potential and the ammonia oxidizing archaea, which makes sense because their niche is a low end niche typically. They're more efficient at using the ammonium when it's not readily available. So the take home for all of this is that these particular biological indicators of soil health lend themselves, I think, very well to organic systems, and that much of what we're doing in these organic systems with animal manures has potential to cross over into other sustainable managements. And most of us that have worked in organic know that we're also going to be working in what we call more conventional systems too, and that there's crossover back and forth, and there's should be crosstalk. So again, nitrification potentials and looking at gene copies of specific or key species of organisms can provide us a sensitive indicator that tells us something about fertility and soil health and perennial and annual systems. And it gives us information about inherent properties and variant properties that we have control over within a season or within several years or even decades. And so I would definitely recommend using more biological indicators, although they tend to be more ephemeral. You can see that in these instances that as long as you handle your samples well, that you can get good information using these biogeochemical processes, and that we can use them to reduce the potential loss of reactive nitrogen in organic and other systems. So with that, hopefully there'll be some time for questions.
Great. Thank you, Dr. Fortuna. That was excellent. A lot of information in there. I know I have many questions about nitrogen management and use of these as indicators. I'll check the chat really quickly to see if others have questions. But maybe before jumping into chat, just want to ask if you have any perhaps overarching, very broad nitrogen management recommendations for organic producers, be it about nitrogen fertility source or, like you mentioned, variant properties that are within our management scope. Do you have any recommendations for organic growers who may be struggling with nitrogen management?
Sure. So I think it's really critical to use combined sources, because if you're just relying on green manures, which a lot of the traditional organic growers did because many people were vegetarian and, Diet for a Small Planet sort of drove part of the movement. But it's very hard. You have to take and if you have animals, you can on site, you can transport that into the system along with your green manures, and the C to N ratios of those components and the chemical resistance or availability varies, and you can mix the two together to get a more ideal C to N ratio. And if you have some humic components, that can tie up along with the mineralogy, some of that available nitrogen. And so something I didn't present was the research of one of my former students, Arnaud Bomick, and he showed that the additions of the compost long-term build up the carbon fractions, and that enables you to capture some of that ammonium that is released, or even nitrates from plant tissue, because plant tissue in legumes has high nitrates too. And that will capture it for a few weeks, kind of similar to some of these, nitropyrin type phenomena. And then it's still available and can be re-released closer in timing with the uptake of the plants. So what we found is if you don't have livestock, you often have to take and import nitrogen and some type of animal nitrogen or biosolids, which some organic producers are still very negative about applying, even though it is okay under the organic standard under certain circumstances. So I would say that's the big take home. So people use fish emulsions, which is really expensive, or feather meal. And if you can integrate animals into your operation and use the waste, that's invaluable. The problem with using the animal waste is the phosphorus buildup. And on that bioreactor study, and one of the reasons why I put it in there is I was hoping I would get some questions about things like this. There was a chemist who was able to extract some of the phosphorus and make struvite, which is similar to, phosphorus fertilizers. It's nice granular stuff. And so I think that's the other aspect that we have to look at. We have to try to figure out how to remove some of that phosphorus from the animal waste and maybe hold it until we get to the point where we've depleted it. And so it's just balancing, I would say, not just the CN, but the P ratio of your system. And then the other thing that could build up would be micronutrients. So just making sure if you're raising chickens and you're giving them copper supplements and things like that, that your micro nutrients don't build up to a point where you might have some problems with legumes or something. But that's all manageable, I think, if you think about doing a mass balance of your nutrients over time.
Yeah, absolutely. And out of curiosity, do you think it's possible to try to balance P and N application by using legumes to reduce the amount of N applied from manure so that perhaps you can meet your plant's phosphorus needs with the manure and nitrogen needs with the legume if both precede the crop of choice?
That's a good point. Because even though legumes have a higher phosphorus requirement in order to do have the symbiotic relation with the nitrogen fixer, that is one strategy, yes, that many people do use. So that's a good point.
Sure. Yeah, but I hear you on trying to minimize P over application, and the struvite thing is very interesting.
Yeah, and another thing about now, if you just have green manures, that we find is that if you've got a mixed cover crop of some sort, or maybe you can do strips of different crops, is that you don't lose as much reactive nitrogen. So you've got something that has a C to N ratio that's closer maybe to 20. When you have like rye with a hairy veg, that's a really simple mix. And then that will reduce the amount of nitrous oxide loss too, because there's a lot of nitrate already in the legumes that is released when the cells senesce, and then you get the ammonification and you get the ammonium in the system too. So I think having mixtures where you do use the legumes, but you also maybe couple them with some plants, like the rye is a good scavenger crop over the winter, and it retains the nitrate and it grows bigger, and then the legume takes off. And so when you turn it over, you get, instead of instantaneous release, you get a little bit of a lag, and that hopefully will bring you closer to the time of maximum uptake. I have some research, but it's pretty old that shows those types of interactions too, where we grew mixtures. And now what I'm doing in not organic but sustainable grazing systems is growing mixed cover crops. And so that extends the gap in the forages that we have. So if you want to finish the animals and sell them off a little later, that might be a benefit. And you have something for them to eat. So you're just not spending a lot of money on hay and other supplements. And so we're using, traditional cover crops as forages, and we are fertilizing them, but we're doing it minimally, and that's still retaining our nutrients. And then we have the below ground that remains. And so you can do that type of mixtures in vegetable cropping systems that have a high nitrogen requirement as well. So I think there's some neat crossover, and I think integrating animals can be very valuable and provide you with some nutrients that you can combine with other things. So hope that made sense.
Does to me. Yeah, thanks. So one of the questions from the attendees, we did not speak to this during your talk, but I'll ask, just in case you have any experience with the potential effect of nanoplastics on basically soil health and plant health, potentially affecting uptake of nutrients, good or bad in this case. I
wrote a book chapter with my another former colleague from North Dakota State, Achintia Bezbora, and I addressed that a range of nanomaterials. So with the nanomaterials, sometimes they're coatings, but the coatings tend to be things that we find like chitin or alginate in nature. So those can work as a slow release, releasing nutrients a little later in the growing season. But then some of the technologies, you have to be careful again that you maintain the balance between the different nutrients. So nanomaterials in many instances are taken up in excess. And so they could, if we're not careful about balancing the nutrients, disrupt the ratio of another nutrient. But again, apart from the size, the concepts behind using the nanotechnology have been around in agriculture, I would say for centuries. And so it's really how long they stay small and not aggregate. And also, those mineral interactions that I talked about that influence, things like ammonium, they also influence what happens, the fate of those nanomaterials. So the plastics can be, sorbed into the organic humic fraction, which we didn't talk about, or they can be sorbed onto cation or anion surfaces. So if they're cation surfaces, it's probably more the mineral component. And so over time, they're going to be tied up as a traditional sized element would be. But with the nanoplastics, I guess it depends on what the carbon chain looks like. So if it's a straight chain, then probably microorganisms could degrade it co-degrade it and maybe use it somewhat as a nutrient source. If it's a more complicated molecule, then it's going to probably persist longer. So if it's what they call a xenobiotic, that would be something that's made by people that organisms haven't seen before. So you're going to have to try to degrade it by putting something into the system or having something in the system that they can use as an energy source, and then accidentally co-metabolize the others. So we trick the organism into decomposing another material because it looks like something they use as an energy source. Does that make sense?
Yeah, I love that. That's very interesting. Is there any sort of easy, maybe in-field measurement, or is there a bioindicator for assessing nanoplastics in soil? Or is that simply a laboratory procedure?
Yeah, that's a good question. So when we look at remediation strategies, they oftentimes use organisms as indicators of toxicity. I mean, so in water systems, we use things like Daphnia, water Daphnia, and you can use microorganisms in a similar way. I can think of one example or a few examples of things like rhizobium populations being used as indicators of toxicity, because that's something that's a beneficial organism. And so there are a number of, they call them PGRPs, growth promoting microorganisms that are used as indicators already for things like heavy metals. And so ideally, you would want to pick an organism that would have a pathway, an energy pathway that would be connected to whatever the material is that you were concerned could have toxicological effects. And so that's what I would do. I would try to look at the literature first and see what people were using and, build off of that. And if I didn't have an indicator, I would try to look at an organism that had a specific metabolism that would have, fairly quick reaction to whatever it was I thought might be toxic. And then you would follow that out over time to see if that organism could compensate or not. So.
Got it. That makes sense. Thanks.
Okay.
Yeah, no, that's great. I also want to jump back all the way to the first project that you presented with the different cation exchange capacity type soils. So with the understanding that like smectites are incredibly high CECs, especially compared to, so we're down here in the southeastern US, we've got these old soils with low CECs. Is it safe to assume that the ability of a soil's ability to absorb ammonium, like you say, is just directly correlated with CEC?
It is, yes. Now, so in altisols, I think most of the soils in Clemson are going to be altisols. There's a lot of iron and aluminum oxides, particularly the iron oxides, and those have variable charge. So their variable charge is dependent upon pH, and you can use that also. So the bottom line is you're not going to be able to absorb as much ammonium because the CEC, even on those iron oxides, is pretty low. And there's not that much organic material either. So you're going to have to account for that so you can get more ammonia volatilization, but the lower pH reduces the potential for volatilization in your systems. So the pH, you can't afford to lime to 7 for most crops. It's just cost prohibitive. So you're probably, about 5.5 to 6, I would say. And I guess for legumes, you need to push it farther up unless you have cultivars that are good at the lower pH. And that's the opposite problem, at high pH, you have micronutrient deficiencies that influence nitrogen fixation as well. So basically, in the older soils, you have the lower pH, and the reduction in the pH reduces some of the volatilization of the ammonia, but there's far less exchange sites. So you I would assume overall have greater potential for loss of ammonia due to volatilization.
Mm-hmm. Yeah, it seemed that way, especially in that that third soil that you presented with the. It seemed as if that would have a lower potential for that, given its CEC. Great. Well, thank you for answering that question. Let me just see if there's any other questions in the Q&A or in the chat. Okay, none over there. Any suggestion for, so as maybe a final question about project number 2, it seems like it may be difficult to balance those two types of losses, nitrous oxide versus ammonia. Do you have any suggestions for growers, just to reiterate again, for striking a balance there?
Yeah, so again, it would be looking at where your carbon and your nitrogen is coming from and what your nitrogen is bound to and how readily decomposable or available that is. And also your weather conditions are pretty critical. You have to have a certain amount of moisture and a high enough temperature. So you have to time your inputs such that you have a more optimum C to N ratio. So maybe not super low if you're applying it before your plant is at a certain stage and can take it up. But, again, your weather conditions are going to dictate part of that too. And if you're not in an irrigated system, then you're going to also have to take into account when you have enough moisture and that you don't take away moisture from your cash crop, the vegetable. So in that organic system, we had drip irrigation, and some years we had to use it, and other years we didn't. So again, I think just trying to really think about how your nitrogen inputs, not just the C to N ratio, but the chemistry and whether it's humified or not, or parts of it are humified or not, and then how that relates to your climatic conditions and whether or not you can manipulate moisture in particular. And so again, if that also reduces losses of the reactive nitrogen and the potential for nitrous oxide emissions, because a lot of people say that in conventional, I don't like that term, no-till systems where we're in mostly monoculture or just maybe a corn soybean rotation, that when we reduce the loss of CO2, that we increase nitrous oxide. But in these organic systems where we have a more diverse amount of substrates that are less or more available, we seem to be able to tie up a lot of the reactive nitrogen in the soil, and that reduces both CO2 and nitrous oxide losses. And I have some papers with my student, Arnaud Bomek, that show that. And he's doing some of that work actually in North Carolina, and what is it? It's the A&M and A&T, right? North Carolina. Yeah. So anyway, you should look him up. Dill has and knows him. And so it's more complicated. But I think most organic growers are very committed and they're very intelligent, and many of them actually have advanced degrees and know a lot about biochemistry and microbiology. So they're fun, but a challenge to talk to because they know a lot and they can call me on my stuff, which is good.
I would take this opportunity to thank Mark Dempsey. Thank you so much for moderating it. It's wonderful as always. And Dr. Fortuna, thank you so much for, I don't know, everybody knows Dr. Fortuna is one of my colleagues at the North Dakota State University. And thank you so much for coming and sharing your wisdom and about the organic soil health and everything, how we can manage. And thank you so much. And